The overall goal of the following experiment is to isolate human stem cell colonies in a quick and efficient manner. This is achieved by culturing human embryonic cells into a dense layer of mouse embryonic fibroblasts. Over time, expansion of the stem cell population increases the number of human stem cells, but also allows them to create island-like colonies in the MEFs. Next, the MEF layer is removed by aspiration, leaving behind the individual colonies of human embryonic stem cells. Results of immunohistochemical staining for stem cell markers show that this is an effective isolation method for eliminating feeders and maintaining colony integrity.
I first had the idea for this method when co-culturing cells in our lab. I wanted to save the stem cell cultures from the overconfluent culturing systems. So the problem that most people will experience with this system is lack of human stem cell culture experience.
We found this technique particularly useful when we need to isolate large numbers of human embryonic stem cells for experimentation.
Compared to other methods such as sorting or hand-picking stem cell colonies, this method is faster and more efficient for isolating colonies from high-density cultures. The mouse embryonic fibroblasts should be treated with mitomycin C or irradiated prior to co-culturing with embryonic stem cells to keep them from going through division.
2 hours prior to MEF seeding, coat each 60 mm plasma-treated culture dish with 2 milliliters of 0.05% gelatin.